Good morning. I guess morning is difficult today because it's raining. And yesterday we had a very nice tower in Krakow, so I think we would better all just relax and have more coffee. But, sorry, I will try to take you through a very general overview about learning and plasticity in biological systems. And I always want to try to link to artificial intelligence because I started in artificial intelligence and moved to biology, computational neuroscience, and now it seems I'm slowly drifting back towards brain-inspired computational algorithms. And I would like to ask you, so how many are you coming from pure biology? No one. To pure biology, yeah. And thank you. And how many of you are coming from computational theoretical neuroscience? Yeah, it's like 30% out of I don't know how many. And how many of you are coming from computer science, machine learning? So we are a very mixed crowd. So something for you will be very. Sorry, what's wrong? So something for you, it will be very familiar, but not for others. So I will try to integrate all that knowledge, and probably it won't be very deep because of lack of time, and I promise to finish much earlier. Okay. So I will start about brain plasticity, and of course, you know, it's for me it's the most fascinating field because learning and memory makes us, I don't know, humans, and makes us adaptive and able to live in changing environment. And basically today I'm focusing on synaptic plasticity, which is ability of a synapse to modify its weight. But of course, you know, that it's also homeostatic plasticity, structural homeostatic. Is it still? Yeah, structural homeostatic plasticity, which refers to modifying dendrites and homeostatic processes during adaptation. But now today we are just focusing on synaptic plasticity. And I will try to make links to artificial neural networks because also I like a lot deep learning, you know, it's boosting nowadays. And in those artificial neural networks, we always talk about synapses and synaptic weights. And if you are familiar with deep learning, you know those different types of learning, like supervised learning and supervised learning. And I didn't put here reinforcement learning, but basically in supervised learning, we have minimization of a loss function, which you sometimes often is error between desired output and actual output. And when we talk about unsupervised learning, so maybe I think you heard about the Hebbian learning, which is coming from biology. So in artificial neural networks, we have synaptic weight. And interestingly enough, in biology, we also have synaptic weight. And it's a very kind of rough form of synaptic weight. It's a product of three quantities. Two of them are presynaptic, and one of them is postsynaptic. And of course, you will see in my talk later, so it can be very, very complex. But now let's start from something very simple. So, synaptically, it's number of synaptic vesicles that have neurotransmitter and also probability of neurotransmitter release. So you might have lots of neurotransmitters, but you're not able to release them. Actually, Dirk is a biologist, so I shouldn't talk too much about biology. And postsynaptically is a maximal conductance associated with vesicle release of neurotransmitter. So today we are talking about synaptic weight in a biological sense, and again we'll try. to neural networks. And of course, you heard about Hebbian learning rule, which tells that neurons that fire together, wire together. And for me, it's very fascinating because Donald Hebb was neuropsychologist or neurophysiologist, and maybe also heard about field neural network, which is used for auto-associative memories, for memory completion and noise removal. So for me, it's really amazing, like biologists tell artificial neural network field how to construct neural networks, and it shows how a lot of potential that is hiding inside our brains. So Hebbian rule is really well known, and Stan somehow is not so famous, but he was talking about long-term depression, which tells that if a neuron is not useful in firing another neuron, so that synapse should be depressed or decreased and eliminated. And finally, we come to our main topic. So it's spike timing dependent synaptic plasticity. So who of you know this phenomenon as TDP? Quite a few, but there are also who do not know. So STDP, it's abbreviation. It's a very kind of recent learning rule, but it's very famous because it was found around 2000, and it just shows how biology, how wonderful and what a miracle biology is, because basically you have potentiation and depression separated just by a few millisecond time, and if you think how neurons and molecules are able to distinguish causality of pre- and postsynaptic activity. Can I point with it? Oh, yes. So, for example, if we have presynaptic activity coming before postsynaptic activity, we have potentiation, so weight is increasing, and if temporal order is reversed, so weight is decreasing. So it means presynaptic activity is coming too late. So for example, you miss a lecture, so you're not useful. So you should be eliminated in neuronal networks. Okay, and here is a temporal difference between those two events, and you see magnitude of increase or decrease is very strong if temporal difference is very narrow. But if you miss, if correlation is basically not existing, so there is no synaptic weight change. So, and people and biologists and also computational and theoretical neuroscientists are wondering how neuron is able kind of to recognize such a short time interval, just 3 milliseconds, we even don't feel what it is in our lives. So a lot of work has been done on STDP modeling and application in neural networks. And when I entered computational neuroscience from artificial neural network, because I was doing learning, so I started modeling STDP, and this was my main topic. And when we talk about models, so of course you've seen already this slide several times, so we can choose different levels of modeling. So you can start modeling from genes, even from genes, and going to molecules, synapses, neurons, and of course when we model brain, and if I manage to show you our recent project, so we model the whole brain, and of course we cannot put so many details as we would wish, but of course we are able to associate it with behavior, with diseases, with cognition. So when you, again, I guess I will repeat, Marielena Linne, so it depends, and also Dieter Jäger, it depends what question you are going to discuss and to answer. So it depends what level you should choose for your modeling. And in STDP models, so I would like to kind of make more a bit theoretical notion. So there are like 3 different big classes of models. One type is phenomenological models, another type is optimal, and finally biophysical models, because biophysical probably is very interesting because we want to understand what's happening inside the synapse. Phenomenological model is very useful if you have abstraction, and if you want to model large-scale neural networks, and optimal models are based on some optimality criteria. And for example, you want some optimal spike timing, or you want some, I don't know, noise reduction, or some function to achieve. So we will go quickly through those types of models, except of optimal, because I'm not so sure I believe in optimality of our brains, but maybe it's a discussion question, so let's skip it for now. So here it's a very simple illustration of a phenomenological model. So basically, synapse and a neuron is just a black box, so we don't care much what's happening inside. We just have presynaptic input, postsynaptic input, not postsynaptic activity, and temporal difference between those two events. And it comes into our black box, and finally we get a weight change. And of course we all want, so our primary goal was to replicate that asymmetrical STDP curve with a very kind of short and sharp transition and around zero when two events coincide. So of course we can use just two exponentials, and that's it, and we don't care what's happening inside. And another type of models is biophysical class of models. So instead of thinking of a black box, so we try to model basically everything what is happening inside the spine, and in neuron, in dendrites, and those models can be very, very complex. So basically approaches that we model calcium dynamics, and those models are based on calcium. Calcium is very important in our lives, it seems, in our brains. And in spines, calcium enters usually through NMDA receptors and through voltage-gated calcium channels, and they are pumped backwards by calcium pumps, and they can diffuse, and they also can be taken by calcium buffers, like here. So all those models are based, phenomenological models are based on calcium dynamics, and of course we have to model calcium. So basically we model calcium current through voltage-gated calcium channels, through NMDA receptor-gated channels, and here is like usual calcium concentration differential equation, and we also can model complex calcium uptake by intracellular buffers, but here is just like a simple decay due to pumping, buffer, and diffusion. And finally, we come to calcium signals, and calcium signals are driving force of plasticity in synapses. So it was observed through computational modeling and also in biological studies that calcium amplitude and calcium time course depends on causality of those two events. So for example, if we have post-pre pairing of two events, so calcium is quite low, and if we have pre-post potentiation protocol, so calcium you see is quite high. And that feature is depending on NMDA receptors, because I guess you know that they are sensitive to local membrane potential and also neurotransmitters. So if a neurotransmitter is coming a bit later than depolarization, so we have quite a high increase in calcium. So it would be kind of very straightforward to take two thresholds, one threshold for LTD and another threshold for LTP, and see how high is calcium, and then kind of infer synaptic weight. So that was done, and it's very famous calcium control hypothesis developed by Schuval in 2005, I think. And here is like, I think this figure is from Grupner and Brunel, another very famous model theoreticians. So basically, calcium control hypothesis tells you that here is calcium concentration and synaptic change. If calcium is very low, so no change, then we have LTD, and if threshold is reached, so we have LTP. And Schuval and his group, yes?
Is it about, like, can we make a connection between this calcium imaging, or this is just a theoretical?
people ask Dirk, but yes, what I read from the papers, but actually I should confess, because my background is more mathematics and computer science, I always find difficult to read biological papers. That's why you have to collaborate with experimentalists, really, that's for sure. If you want to achieve something in neuroscience, you should find your way to kind of to find a lab or, I don't know, collaborator, because for me, biology reading papers, it's a mess, sorry to tell. It's a lot of those chemical substances and interpretation. So, but yes, that is observed also by calcium imaging. Okay, so Cheval and his group applies that learning rule, and you see here is a STDP curve, and are you happy with it? So it's kind of not very asymmetrical, and we have LTD window for positive temporal intervals. It means if presynaptic activity comes before postsynaptic activity, we still have like a depression, which is probably true or not true, but we didn't want to have that LTD window. So it shows you that it's not only calcium amplitude, but something more which is involved in plasticity. And that's why computational neuroscientists together with biologists started looking downstream and investigating what's happening inside. I mean, when calcium activates complex second messenger cascades. So here is a diagram, but probably I will not go too deep into it. So basically when calcium enters through an MDA receptor channel, so it activates calmodulin, and calmodulin activates CAM kinase 2, and CAM kinase 2 is calcium calmodulin dependent protein kinase 2. It's even hard to pronounce it, and we'll show you later. And also calcium activates phosphatases, but it's not here in this picture, and also many more those molecules. But basically, if we see and if we can draw a schematic diagram, so phosphatases are responsible for depression and kinases are responsible for potentiation. And interestingly enough, they are competing, so they are inhibiting each other. To model such complex biochemical reactions, we have to apply differential equations, and usually here is a very simple example, like if you have two species of molecules with forward and backward rates of production of a product, so we can describe it by a ordinary differential equation. So it's like you just multiply those two with a forward rate and subtract product multiplied by backward rate. So basically there are of course more complex equations, but this is kind of like a basic how to model those second messenger pathways. And here is one example about calmodulin. So calmodulin is sensitive to calcium, and when calcium binds to calmodulin, so it performs conformational changes and becomes active, and it can activate CAM kinase 2 and induce LTP. So here is just an example how calmodulin is activated by calcium, and we have such equation, and calmodulin needs 4 calcium ions, so we add such equations on and on. And finally, here is like a simple model, a bit still abstract, about those second messenger cascades. So LTD enzyme is still kind of abstract, and it's sensitive to calcium, to glutamate. LTP is sensitive to calcium. And those two have a linear summation, and we have like a symmetrical STDP learning rule. And those models can become really complex and really advanced. And a lot of those models also are based off CAM kinase 2. And CAM kinase 2 is very important, and maybe if you are interested in learning and plasticity, maybe you heard about like 2 approaches in modeling learning and synaptic plasticity. Like synapse can be on and off state like a binary switch, or synapse can increase weight gradually. So, for example, in artificial neural networks, synaptic weights can grow continuously or decrease. And in biology, I think we should ask Dirk about his opinion. It's still kind of debate if synapse can be in on state and off state, or it can undergo some continuous changes. So that approach of binary synapse switch tells you that synapse can be either in up state or in down state, and it can perform transition from down state to up state or vice versa. And here is schematic diagram how it happens. So if we have a down state and we want to go to up state, so we need a lot of calcium and we need to activate kinases. And if we are in up state, we need a bit less calcium and phosphatase is activated and we go to LTD. And biological mechanism which supports such a hypothesis is a complex holoenzyme called CAM kinase 2. I already mentioned it. So I think it's like a universe. If you think it's only just a single molecule, but it has 6 subunits, and those 6 subunits can live their own life, but they are also sensitive to life of, say, neighbors, neighboring subunits. And basically, CAM kinase 2 can be in active state, phosphorylated, highly phosphorylated up state, at resting calcium concentration and also at high calcium concentration. So it's called bistability because it has like different features for different calcium concentrations. It can have highly phosphorylated states. So here is a model by Grupner and Brunel again, which is based on CAM kinase 2 activation, and you see here are a lot of those complex molecules, and I don't want to go into too much details because I think I won't have enough time, but basically CAM kinase 2 is activated by calmodulin, and it can be inhibited by calcineurin and phosphatase 1. And what is nice, we were able to reproduce at a symmetrical weight change, but of course there were 14 differential equations just instead of a few in phenomenological model. And such models can provide you insight how it happens and why, those synapses are sensitive not only to the amplitude, but also to the time course of calcium signals. So I was a bit, so this model I showed you before, it was bistable, and my previous work, what I did, so we modeled a tristable synapse model, which was based on CAM kinase 2 and phosphatases, and we modeled those molecular pathways inside the spine, and that was very helpful for me to do what I'm doing now, because I kind of have understanding what's happening inside the spine and how calcium is causing those changes, and why it's not calcium amplitude only, but also calcium time course is responsible for synaptic changes and sensitivity to everything, basically, to location of a synapse, to frequency of stimulation, pairings, and duration of stimulation. So those kind of detailed models gives you enough information to move backwards and make phenomenological models. And here is more complexity, so you can add more and more details unless, you're happy with your current model predictions, but basically there are lots of molecules. It's like a space where a lot of things are happening. And as I told, such modeling, when you model biochemical pathways, is very helpful because you can go backwards and construct more phenomenological models. And this is a very nice model, just a single differential equation by Grupner and Brunel, and his group together with Henry Markram just published very new paper this year, I think few months ago. So what they did, so they put all that information which is coming from detailed molecular modeling into a single equation, and basically the system is tristable, and they have a potentiation term which is kind of sensitive and is modeling kinase. They have LTD term which is reflecting phosphatase influence, and some term which is kind of taking place when there is no calcium, kind of active processes are happening. And in the new papers, they showed that this model is able to reproduce variety of plasticity learning curves. And in general, there are lots of models, and what I showed you is just like a tiny introduction. And like 10 years ago, Maria-Elena Linnet, together with her colleagues from Stockholm, also from US, Kim Blackwell is a very well known about on her modeling of biochemical pathways. So they published a review, and already at that time it was like more than 100 models of synaptic plasticity in different brain regions and different, approach, biophysical or phenomenological. And also I checked recently, I think yesterday, so you see, if you put into a PubMed model synaptic plasticity, so you see this year it's above 1,000, no, last year it's more than 1,000 papers. So that is very important if you read the paper to understand where it belongs to, if it's something really very new or if it's derived from previous work. And why so many papers are existing? Because STDP is not so simple as we saw. It's not only like that asymmetrical weight change we wanted to reproduce. It depends on everything. So for example, if we have higher frequency, we have Hebbian learning. Frequency is low, so we have like inverted Hebbian learning, so we can have Mexical head, and it's about hippocampus. It's my main area of research in hippocampus. We have like different areas, for example, cortex and neocortex and inhibitory synaptic plasticity. So all those synaptic rules are different. And I think a goal of neuroscientists is to find like common links between all those different rules and some unification of plasticity rules. And that paper I showed you before, maybe I should go back a bit. Maybe you could check it. I even don't know how to pronounce Hidemni. Sorry for bad pronunciation. And it's with Markram group and Grupner and Brunel. Okay, so STDP is complex, plasticity is complex. And to put more complexity, here is like a short summary what we could talk about for like one semester, I guess. So we already discussed like stimulation protocol, a bit post and presynaptic activity, frequency, but plasticity also, synaptic plasticity depends on dendritic plasticity, because can you imagine your dendrites also plastic? And here is Herman Kunz, and I think he will tell us more. Because it's, again, I'm not a biologist. So dendrites can remember what happened before. So for example, if you induce plasticity in one region, so ion channels become more excitable and they influence synaptic changes that are going to come later in your life. And also they depend on neuron time, brain region, and brain disease. And I am doing also modeling of brain diseases, so for me it's a very interesting topic. And one thing I wanted to mention, it's neuromodulation. So of course you all know what dopamine is doing for us. If we anticipate something nice, so we are doing everything so easily and so efficiently. And so sorry, I may be going too fast, but I'm afraid. So
you can still use like 10-15.
Okay, and here is a very nice, I like that work a lot because it shows you influence of dopamine in your hippocampus. Here we had like conventional STDP rule, if you think, and that is experimental data. And you see, if you have dopamine coming in your brain because you anticipate something nice, so LTD is converting to LTP, so no depression, and also LTP is boosted, so you have stronger LTP, stronger potentiation, and also learning is much faster. So here, for example, when you are not interested, hopefully not here, but of course we know some lectures, we are just looking at your watch and when it will end. And here, if you have dopamine, so you see plasticity is much faster, you don't need so many repetitions. I think I put it not in the right place. Okay, so here how to model dopamine influence. So we can model in a phenomenological way, and also we can model in a very detailed molecular detailed way. So in a phenomenological way, so basically we have some signal which is referred to as dopamine, and that dopamine signal is multiplied by eligibility trace, because you know dopamine is not coming directly after stimulus, so you have to wait like few, I don't know, seconds, even maybe minutes, and STDP is happening on a millisecond scale. So we need, so if we have pre and postsynaptic activities, so we need some eligibility trace, which can be also calcium or some molecules activated by calcium. And once dopamine comes here, so it just increases the influence of that eligibility trace. So that is a very simple way of modeling. And here is a very abstract way how to include in your abstract model. So you have basically weight and multiplied by a third factor, which is dopamine. And that is very useful in neural networks, but if we want to understand, I will go a bit back. So I have a very nice, I don't know if not exactly collaboration, but constant keeping in touch with Jeanette Hellgren Kotaleski from Sweden, where we last year had our summer school. So they modeled all the molecular pathways of dopamine in stratum. And they looked at the influence of cortex activation and dopamine, which is coming from substantia nigra, and they did all that molecular machinery description in mathematical terms. And they were able to show that synaptic weight change is dependent on proximity of cortex activation and your reward activation. So this is really a fundamental work, I think, on a very detailed level, very complex. It was a PhD thesis and a lot of papers. So if you are interested, you can read that reference. But basically, it showed you that if you have dopamine following your cortex activation, so synaptic weights are increasing and strengthening the response. Okay, so let's go forward. Okay, and now we come to one more level of complexity, because usually when we talk about synaptic plasticity, okay, we talk about dendrites, about sinuses, about spines, but we forget astrocytes, and astrocytes are increasingly recognized important in our daily functioning. And Maria Elena will have a lecture, and I think tomorrow, and you will see how important astrocytes are. They are not only housekeeping, they are not only cleaning your brain, but they are implicated in synaptic plasticity. And that is one of our model, we tried to do it. So I will skip it, and Maria Elena will tell I hope so, a bit about molecular machinery, how astrocytes are not cleaning your brain, but contributing to neurotransmitter release and modulate synaptic plasticity. So we have lots of differential equations and lots of parameters, but kind of we managed to replicate experimental data, and it gives hope that our work is not it's not okay, I will skip it. And finally, we talk a lot about how neurons learn, but we never talk how we forget. And just think forgetting is important. So it's only about how much we can learn, but not how much we can forget. So I think last year I got a lot of questions not about learning, but about forgetting. And I should confess I'm not an expert in forgetting and really bad in forgetting. But basically neurons forget, and it's also very important. And how it happens. So few main hypotheses and proven by biologists are that AMPA receptors, they move from the surface of membrane and the synapse becomes weaker. Synapse can shrink, so the volume of spine becomes smaller and it undergoes also depression. And also here is a very nice paper, but I didn't put a reference. I think it's about a special protein kinase MZ. So it's involved in maintenance of your memories, potentiated synapses, and if you kind of inhibit PKMZ, so all your weights, all your memories can go down and you will start forgetting. And in principle, if we talk not about molecular mechanism, but you see how important it is to understand molecules. Because we can talk about general, so if we have nice life, interesting life, enriched environments, of course you more faster forgetting what's happened before. So enriched environment, so all the memories are not so strong anymore, also ongoing activity, and recently it was shown by studies with humans that during REM sleep, synapses undergo depression. So here is like also a very nice paper if you want to read it. So those green synapses are like your new memories acquired during the day. And during REM sleep, so those green synapses are going down, so kind of depression is taking place. And that is very important if we move forward, because forgetting is important not only for healthy functioning, for increasing your memory capacity, like in artificial neural network, but forgetting also is impaired in various neurological and psychiatric disorders. So, for example, post-traumatic stress disorder is, of course, you know what it is. So, if you have a very stressful event, so it's kind of flashes back and interferes and causes panic attacks. So, it's impaired, forgetting is impaired in depression, also anxiety. So I also read very interesting story about post-traumatic stress disorder, because I am teaching psychology students and we often discuss. So it was suggesting that if you have a very traumatic event, so it's better to stay awake and not to sleep too long, because if you stay awake and I mean, so synapses cannot be potentiated well enough. So there are a lot of theories, and forgetting also very important, not only in humans, but also in artificial intelligence systems. And I just put one slide here because I also found a very nice application that if you have a neuromorphic devices that are going online and have online learning, so they can kind of weights can saturate and they can lose capacity of learning, so they should kind of sleep, quote unquote, whatever that means, and sleep restores normal learning. So inspiration from humans and biology can also be transferred to artificial intelligence algorithm. And here is also a nice paper, backpropagation and the brain. So if you are interested in linking those two fields, so please read that paper. But I will skip it for now. And finally, I will show you a few slides about my own projects. So I'm involved in modeling synaptic plasticity and learning in hippocampus in Alzheimer's disease. So we basically Okay. Have a very nice consortium of experimentalists. A group in France is doing experiments on toxic peptides in Alzheimer's disease, and together with my colleagues in Italy, we are modeling those effects on neuronal level, and also we are trying to put into a bigger scale network. Colleagues in France investigating toxic beta amyloids, eta amyloids, and ACD, which is like also a toxic amyloid. And those are accumulating in the brain, in cells, and they are forming, for example, beta amyloids are forming plaques, and those plaques are observed in hippocampus, also in cortex, and then they impair memory and actually ability to potentiate synapses is lost. So we were building a synapse model and also kind of putting all those toxic effects on synaptic level, and also we have microsecond, and we could replicate the results of those toxic peptides. And our long-term goal, I don't know if it works. Is to put into a large-scale neural network. So here is a hippocampus, the whole reconstructed hippocampus, and you see activity in those neurons, and it's done together with EPFL and hippocampus team in Italy. So that is one project. So how many minutes do I have? 2, 3. Okay, so I will skip. I will skip also slides about consciousness. We had a very nice conference about consciousness 2 months ago. It was like a big event, Human Brain Project and eBrains. And we were discussing different aspects. And here is just a slide for you to think if memory and learning is important for us or not. And Clive Waring is a resident in UK, so he had herpes simplex, I think, and that virus damaged his brain and hippocampus, and he can remember only like 7 seconds. And he was interviewed, and you can find on YouTube his interview. He plays piano wonderfully, but he says that it feels like being dead. So without memory and without ability to travel in time backwards and to imagine yourself in the future. So it feels like being unconscious, like being dead. So that was like a philosophical, as you see, so from molecules we can go to learning, to consciousness, to artificial intelligence. So many levels we can capture with mathematics and informatics and physics. Okay, and also I collaborate, so 1 more minute, okay. I collaborate a lot with our neurosurgeons and neurologists, and moving a bit towards translational neuroscience. So when we discuss consciousness with neurosurgeons, so they think, okay, we use Glasgow Coma scale. If a person cannot feel that we are touching, if a person cannot open eyes, so we think a person is unconscious. So, but what about consciousness in a broader sense? If we don't remember ourselves, so are we conscious or no? So that is like a food for thought. Okay, final remarks. Maybe you can kind of make final remarks yourself in your brain, and I will skip it. And here are a lot of open questions, and for me I think the most fascinating thing is like going through different levels and putting all that knowledge for the benefit of people, patients, for treating diseases, and of course afterwards coming all those, I don't know, artificial intelligence algorithms and everything else. And finally, okay, here is my country, you see that's yellow. So here it is, very close to Poland, and Kaunas is in a center, so I work here. And finally, also want to show our university. So we have a fundamental Science in Neuroscience Institute. We have a very strong University of Health Sciences, and we have a very huge clinics with everything basically. And recently we started doing, it's final slide, project with e-brains, and you will be studying e-brains, I think, tomorrow. And we are trying to predict the outcome of neurosurgical treatment of Parkinson's disease, because you know that electrode goes inside the brain, and sometimes there are complications, both motor output and psychiatric. And together with neurosurgeons, and together with Professor Victor Yirs and Marcel, we are trying to predict what's happening after the surgery. And here are my collaborators, Maria Elena Line, 10 years, maybe more, 15, and also Bruce Graham. Yesterday you had a tutorial, so he was my also teacher, and I learned a lot from him, and Jeanette from Sweden, Tina from Maria Elena group, and Michele Migliore from Italy, my students, my physicians, neurosurgeons, human brain projects, councils. And if you are interested what we are doing and you would like to visit our university or to have a shared project or whatever, so please email me, and I hope you to welcome in our country. So thanks a lot.
Thank you, Ausra, for the very interesting talk. And I think now you were actually exactly 45 minutes, so
yes.
We are like 5 minutes late, but I will take it into account. So now we can take maybe 1 or 2 questions.
Hi. So I was wondering a little about forgetting, and specifically healthy forgetting, because you mentioned at some point, I think, about like an excessive kind of stimulation kind of interfering. And I think we need to separate between forgetting and not encoding memories. But I was wondering whether we need to specify a mechanism for forgetting specifically, independently from simply rewiring from learning that interferes with prior wiring.
Yeah, I was forgetting, as I told, I'm not expert in anything, I guess, but particularly in forgetting. And it's hard to say. I mean, it's also as complex as learning forgetting is, and it's also rewiring the brain. It's like molecular machinery involved, and also endocytosis, and also glutamate release. So I think it's a very complex mechanisms, and it's not well understood yet. And I think it's not well kind of investigated, and it should be focused also more on this field, because also it's, as you said, it's connectivity, and I mean, everything is involved. Yeah, so I agree, and I told you that I receive questions not about learning, but about forgetting. But forgetting, as you know, it's very important also in artificial systems. Like if you overtrain a neural network, so it loses your ability to generalize. Yeah, so I'm not sure if I answered you at least a bit your question. Yeah.
Yeah, let's take one more question and then we will move on, unless there are questions in the online. Okay.
My question is regarding the binary sort of nature of weak and strong states in learning. Is it just convenient to categorize it like that, to model or to explain, or do you really believe there is a up and down state?
I think if you have like a binary approach to synaptic weight, so it gives stability to your like memory maintenance, because small disturbances wouldn't cause your memory fail. And for example, if you have like decrease a bit in calcium, so that CAM kinase 2 or bistable approach would allow you to maintain memory, and it wouldn't be so sensitive to environment and to noise. So that's basically benefit for biology, but for modeling, it's more complicated to model binary approach. Because you have to design a system which is bistable or tristable, and it's a bit more complex and gradual increase of weight with some weight like maximal or minimal range. But I think it's in biology we have all that stability, ability to function in noisy environment, in unexpected environment, so binary approach gives you stability.